Over the last 100 years or so, the Bible has been under a prolonged attack by its critics, and especially recently it's been criticised by a number of scientists as it disagrees with the widely accepted viewpoint of the origin and development of life. And that's one reason why many people have now turned their back on the Bible and consider it is irrelevant in the 21st century. To many in the outside world, the Bible is seen as a fable. It is seen as a primitive explanation of the world based on superstition, and in today's society it is seen as completely out of date. Those of us who still believe in it are simply clinging on to an old story from a previous age, and an old-fashioned ideal that's got no place in today's world. Science, on the other hand, offers an up-to-date, rational viewpoint. It is impartial and deals only with the facts, which makes it very difficult to argue against. Scientists make very compelling arguments and appear to destroy the teachings of the Bible about the origins of life and the universe. We trust scientists because they know what they're talking about. We use computers and mobile phones every day, and they work because scientists have designed them based on their understanding of how the world and how the universe behaves. When we drive our cars over bridges, we trust the fact that scientists and engineers understand the properties of materials and have calculated the stresses and the loading capacity to ensure the bridge will not collapse. We trust doctors and chemists who devise new medicines and vaccines for various diseases. And over the past century, scientists and engineers have achieved some truly remarkable things that would seem impossible at the start of some of our own lifetimes. And so we trust scientists. They work with facts and with logic, and it has got the human race a long way. So when scientists say the Bible is wrong, people trust them. And why not? They seem to be right about everything else. Not only that, but the vast majority of people in the world do not have any scientific or engineering training. So we're not in a position to disagree with scientists. They're the ones with the knowledge and the expertise. And just look at what they've achieved. So people assume they must be right. Well, scientists work with facts. Strictly speaking, they work with evidence, information they have observed from experimentation. And from this evidence, they put together theories about how things work. And then they test those theories to see if they stand up to scrutiny. Well, the aim of this talk is to provide you with evidence and with facts, the same information that scientists have. And we'll have a look at the conclusions that the scientists have come to. And you might like to consider if you agree with them, or in fact if you might not be quite so ready to accept everything that scientists tell you, especially when it comes to the questions surrounding the origin of life. Now, what I'm not going to tell you is that the scientists are wrong and the Bible is right. In the spirit of this talk, the evidence you decide, I'm just going to give you the evidence and you can decide for yourself. Now, to do this, we do have to get quite technical. Now, hopefully it won't be so complicated, it's just going to bamboozle everybody in the room. Some of it is quite complex and it may be difficult to understand. And I'll try and make things as clear as possible. What I'm going to focus on today is the origin of life and the complexity of life, and see how this fits in with scientific theories. And even if you don't understand everything this evening, the thing I hope you will appreciate is the sheer complexity of life. Now, scientists would have us believe that the universe started spontaneously in a big bang. And then over millions of years, life on our planet has evolved. And this is what scientists tell us based on the facts as they see them. And from this, they assert that the Bible's account of the Creator must be wrong. But when and how did life itself start? When we talk about evolution, we're not talking about how life started. Evolution assumes that life has already started, and it gradually changes from one thing into another. But let's start with the more fundamental question. How did life start in the first place? Well, scientists have asked this question, and a lot of time and research has gone into answering it, or I should say attempting to answer it. the general consensus among scientists is that life somehow started spontaneously. That's the only option available to us if we remove the possibility that life was created by an almighty God. So scientists have tried to find the conditions that would have led to the spontaneous creation of life. In the 1950s, two scientists, Stanley Miller and Miller-Urey, working at the University of Chicago, came up with an experiment, a very famous experiment, now called the Urey-Miller experiment, and I remember being taught about it in school myself. And the experiment tried to mimic the atmosphere of the early Earth, and it's taught as part of some GCSE science curriculums today. Now these two men said, what happens if we take a mixture of chemicals, a prebiotic soup, if you like, something that might have existed upon the Earth millions of years ago, assuming that were the age of the Earth, say water, hydrogen, methane and ammonia, and what happens if we pass electricity through it to simulate lightning storms that might have been going on the Earth millions of years ago, assuming that was the age of the Earth. Could that produce life? And they set up an experiment to do just that. They set up some equipment which included two chambers that were joined by tubes in a circuit. One chamber contained water, representing the ocean on the Earth, and it was heated. And the water vapour moved through the circuit into the other chamber, and it was then cooled by a condenser as it travelled back to the first chamber. And a trap at the bottom allowed water and chemicals to collect and sampling probes to extract material. In the main chamber, they put these gases, ammonia, methane and hydrogen, and of course there would be water vapour from the water. And they then discharged electrical sparks in the main chamber via a couple of electrodes to simulate lightning. And they set all this apparatus going, and they left it running for a couple of weeks. And after 2 weeks, they looked at the chemicals that were created. And to their surprise, they found that over 20 amino acids had been created. And amino acids, as you may have heard, are often referred to as the building blocks of life. Once amino acids are created, we can build proteins from them. And proteins are used to build cells and control the processes that go on within the cells. So amino acids are essential for life. So scientists had struck gold. A very simple experiment had demonstrated how life could have spontaneously come about upon the Earth, and since then other scientists over the last 5 or 10 years have repeated and elaborated upon this experiment and produced other chemicals that are needed for life. And once we have life, we can then use evolution to explain how different forms of life have developed. So there you have it. Life is not so difficult to create, if the conditions are right, which they might have been on the Earth. And it doesn't need any intelligent intervention or design in order to come about. These were random reactions, and yet these building blocks that lead to life were created. Well, that's how the scientists like to portray it. And if that were the whole story, I would just pack up now and we'd all go home. But let's wind back a little and have a closer look at this experiment and the deductions that were made from the results. When Miller and Urey came up with this experiment, they made some assumptions. And all good scientists will state their assumptions when they write up the results of their experiments. Their main assumption in this experiment was the composition of the atmosphere. They came up with these gases, hydrogen, water, methane and ammonia, by guessing. They didn't know these were the gases that were around many years ago. In fact, no one knows what the gases were around. Maybe they're the same gases that we have in the atmosphere now. But Miller and Urey knew that these gases were the most likely to generate the right kinds of chemicals these gases might produce amino acids. So instead of finding out what gases were on the Earth and how they might be used to make life, they worked in a sort of backwards way. They knew the answers they wanted to get, and they came up with an experiment that was most likely to produce those answers. Now, that's fair enough, but we need to be very careful about what deductions we can draw from an experiment that's been constructed like this. We cannot, for example, say this proves the Earth was made up of these gases. The composition of the gases is still an assumption, an educated guess. Do we have any proof that these were the gases on the Earth? No, we don't. Let's just take a look at the gases again. We've got hydrogen, we've got water vapour, we've got ammonia and methane. What gas is missing? What might you expect to see there? Anybody? Sorry?
Nitrogen.
Nitrogen? There's no, well, there is nitrogen there as part of something. There's another gas. It is actually there, but it's not in a free state. Anybody else?
Oxygen.
Oxygen. There's no oxygen. I mean, technically, as I said, it's there, it's part of the water, but there's not in gaseous form. So why wasn't oxygen included in the experiment? Because oxygen, in its free state, like we have in the atmosphere today, is like a poison to amino acids. Oxygen reacts with amino acids and breaks them down. If free oxygen were present, like it is today, then the amino acids produced would have been destroyed as quickly as they'd been created. And so what has the scientific deduction been? Well, scientists therefore assert there must therefore have been no oxygen in the early atmosphere. Now we've got to be careful here. This is a deduction based on an answer that we want to get. It's not based on any observed scientific evidence about the Earth's early atmosphere. It is a deduction based on an assumption. Scientists believe that atmospheric oxygen was produced by plants millions of years after the start of life. And notice I said there that scientists believe, because that's what it is. It's not based on scientific fact. Now maybe that doesn't matter. So what if there was no oxygen? Well, if there was no oxygen, then that gives us a problem. We've probably all heard of the ozone layer. There's a layer of ozone in the atmosphere that protects us from ultraviolet rays from the sun. Without the ozone layer, the ultraviolet rays from the sun would destroy all life and the planet would be completely dead. So without ozone, there would be no life. But what is ozone? Anybody know?
O3.
O3, yes. It's oxygen. Most oxygen molecules in the form of O2 is O3. Just hold on a minute there. We were told that there was no oxygen in the atmosphere because life cannot form with oxygen present. But then we're told life cannot survive without oxygen being present. So life can't start with oxygen, but it can't survive without it. How strange is that? We might argue this would indicate life cannot come about spontaneously. Maybe it points to an intelligent creator. So how do scientists explain this? Well, we simply modify our theory. In order to be protected from the sun's harmful rays in the non-oxygen environment, perhaps life began under the sea. Perhaps near the hydrothermal vents that are found at various points around the globe. So these are seen as candidates for the origin of life. But how the amino acids would have come about here is not known. Scientists haven't suggested any mechanism. Perhaps it was somehow made under the Earth's surface and then injected into the sea. The only trouble is, amino acids in water undergo a process called hydrolysis, and the oxygen atom in the water molecule prevents amino acids from linking together, and eventually they are broken down. So over a relatively short period of time, any amino acids would be completely destroyed. And also, the temperature of these vents is very high, and organic chemicals are highly unstable at these temperatures. The amino acids would just decompose at these temperatures. So let's just stop and think about the evidence that we have. Based on scientific facts, that's what scientists work from, based on the fact life cannot have occurred naturally on the surface of the Earth if atmospheric oxygen was present. But life could not have survived on Earth if atmospheric oxygen was not present. Life cannot have been created under the sea because water would have prevented amino acids from being preserved. The facts lead us to consider another possibility, that intelligent intervention is required in order to create life on the Earth. And in fact, that is precisely what the Urey-Miller experiment showed. The textbooks don't always include all the details, but when Urey and Miller set up their experiment, if they wanted to demonstrate there was a chance that life could have started spontaneously, then what they should have done was set up the initial conditions of their experiment and then leave it to run its course. They couldn't interfere. If they interfered, then they were introducing intelligence into the process. That would completely invalidate the purpose of the experiment. And what did they do? They interfered. They had to interfere. In the experiment, the amino acids were created in a special reservoir which contained a trapdoor. And Miller-Urey extracted the amino acids as they were created and stored them in special vials. And why did they extract them? Because they knew they would not survive the conditions of the experiment. But furthermore, there's another piece of information we need to be aware of. Amino acids are molecules that have a certain shape. But in fact, for any given amino acid, there are two shapes that it can have. Amino acids have handedness, sometimes it's called chirality. And if you look at my hands, they're made up of the same elements. I've got 4 fingers and a thumb on each hand. But they've got different shapes. And it doesn't matter how I turn my hands, one is always going to be a left hand and one is always going to be a right hand. I'm never going to make one look like the other. Now, amino acids are the same. They have a right and left-handedness. And all reactions that create amino acids create a mixture of left and right-handed amino acids. And so, unsurprisingly, the Urey-Miller experiment created a mixture of left and right-handed amino acids. But if we look at the amino acids in every living creature, the many trillions of cells, the many gazillions of proteins, they are all left-handed. There's not a single right-handed amino acid. Interestingly, sugar molecules also have handedness, and if we look at all sugars that occur in living creatures, they're all right-handed. So once these amino acids have been created, some mechanism needs to be in place to separate and to extract the left-handed ones in order to produce the proteins required for life. If a right-handed amino acid is used in a protein chain, then that protein is no good at all. We'll see that a little bit later. So what undirected, accidental process produces only left-handed amino acids? Well, there isn't one. Scientists have come up with suggestions of how in a reaction you might get a few more of one than the other. But they're only small imbalances. And don't forget, it must have come about accidentally, without any intelligent intervention. Now, some might say that the evidence suggests an intelligent creator. It looks like some intelligent process has taken place to organise things. Things do not simply go from a state of disorder to a state of order. What does the Bible say? It says, in the beginning, God created the heavens and the earth. Now, I hope you're keeping up, because so far we've been looking at the simple stuff. We've been looking at amino acids, the building blocks of life, and we've been looking at some of the problems that are faced with creating these chemicals in a random, undirected, accidental way. And we keep using this term, the building blocks of life. What we're looking at is the individual bricks that make up the building that we call life. And scientists say that these join together to form proteins, as if that was some straightforward process. Well, we're going to stand back now and look at the bigger picture. Let's not focus on the individual bricks. Let's stand back and look at the building. In fact, let's stand back and look at the city that's been created from these blocks. Let's assume that spontaneous creation has occurred. Let's assume that all the problems we've looked at are overcome and left-handed amino acids are created. Well, we now need to join these amino acids together into proteins. I'm going to show you how that's done. And to do this, we're going to look at the so-called simple cell. Now, sometimes when describing evolution, it's said that complex chemicals collected together to form a simple cell. And the impression given is that the cell is a collection of a few chemicals that could work together and reproduce themselves. But the simple cell is far more complex than that. The cell is the basic unit of all life. All living things are made up of cells. And plants and animals are made up of many different types of cells. The human body contains more than 10 trillion cells, and there are about 200 different types. Now, all cells have got common features, whether they're animal cells like ours or a single-celled organism like the euglena in this picture. Euglenas are what forms the green scum on the surface of ponds in warm weather. A cell is alive. It is alive as you or I are. It breathes, it takes on food, it gets rid of waste, it grows, it reproduces, and in time, it dies. Now this picture shows the main parts of a typical animal cell. And just like a house has got a kitchen, and a living room and bedrooms and a bathroom, a cell also has specialised areas which are partitioned off for separate uses. And these areas include things like the mitochondria, which produces the cell's energy. We've got the nucleus, which controls the cell's growth and function and reproduction. And it contains the DNA, the cell's source of information. We have something called a lysosome, which is the cell's waste disposal unit. We have the Golgi apparatus, used to store and transport proteins. We have the endoplasmic reticulum, which processes proteins. And in all, there are more than 20 different sections of the cell. There's going to be a test afterwards, by the way, just to see how much you remember. And the cell is continually making new proteins and getting rid of old ones. And we're going to have a look at the manufacturing and transport process for proteins. We're going to follow the journey of a protein from the nucleus to the lysosome, a distance of about 0.002 millimeters. And the lysosome, if you remember, is the cell's waste disposal unit. So when the cell detects that it needs more of this protein, then this manufacturing process is set in motion. And every protein has the information for its manufacture stored in the nucleus in the form of DNA. And you've probably all heard of DNA. It's this great long double helix molecule made up of millions of smaller molecules. And it's all folded up and stored in the nucleus. And that DNA, effectively, is the blueprints for making whatever creature it sits in. So it defines everything from making the proteins to it defines the colour of your eyes, the height you're going to reach. All that's described in the DNA. So if you watch that happening now I'm going to show you a video, and hopefully it comes up okay. It's a bit dark to start with, but it'll come up. We're going to go into the nucleus of the cell. So there's the nucleus here. As I say, it gets a bit dark here, but hopefully you can see it. There we go, you can see it. We're entering into the nucleus. Now you notice there's lots of holes. Now these holes are like security gates. They'll only let certain things in and out of the nucleus. So if we go to the nucleus, and we can see our DNA, this double helix. And what we need to do is find the section which describes the protein that we need. We look along this DNA. We then find the section which describes the protein we want to make. Once we reach that point, there's a little molecular machine that comes along and effectively unzips that part of the DNA. So it's unzipping that particular section. Here's the bit of protein we need to make. Another molecular machine then comes along and makes a copy of that section of the DNA. In fact, it doesn't make a copy, it makes a sort of mirror image of it, and it's something called RNA. And it's just a small portion, a mirror image of the DNA portion. And a security marker is placed on each end of that RNA. And that RNA then goes to one of the security gates. The security gate scans that security marker. If it's happy with it, it opens up, lets the RNA out of the nucleus. So out it goes. If the marker isn't good, the gate stays shut and the RNA decomposes. Out comes the RNA. It's picked up, or it gets taken to something called a ribosome. A ribosome is like a factory for creating proteins. So here comes the ribosome, and using the RNA, the ribosome now joins together. Now that is an amino acid. And here's another amino acid which is being joined to it. And it's joined together in a very specific order, depending on the coding on the RNA. So this chain of amino acids is in a very specific order based on this coding here. And this chain is the protein. It's not called a protein yet, it's called a polypeptide chain. So here it's being produced. It's coming out of the ribosome. Technically it's not yet a protein. It now needs to be folded in the right way. And that's done in the endoplasmic reticulum. So in the endoplasmic reticulum, this folding occurs. As I say, you can't use the protein until it's folded correctly. Now this is where the handiness comes in. You can see it being folded there. Now if you've got an amino acid which is the wrong handed, if it's right-handed, it'll fold the wrong way. One item will just fold the wrong way, and it'll make that protein absolutely useless. It cannot be used unless it's folded in a very precise way. So once that's folded, it's taken by a vesicle to the next part of the process. Okay, so there's our protein. Now that's the end of that first animation, just to show you where we are in the cell. So the RNA has come out of here, the ribosome has created the protein, now being folded in the endoplasmic reticulum. I've got another animation showing you the next stage of the journey. Now this is in a slightly different style, so hopefully it's not too distracting to you, but it's just picking up from where the last one left off, okay? So the protein is going to be taken to the Golgi apparatus. The Golgi apparatus acts a bit like a post office. It sorts things and decides where things go. So here's our vesicle containing our protein. In fact, in this one, there are 3 proteins in this particular example. And it gets taken to the Golgi apparatus. And the vesicle, when it reaches the Golgi apparatus, it merges with the Golgi apparatus and just delivers those proteins. Now, as I said, the Golgi apparatus is a bit like a post office. So once the proteins enter in, there's a special sugar molecule added to it that acts like an address marker. And that tells the Golgi apparatus exactly where that protein needs to go. So it's given like an address label, and then it gets pushed through the Golgi apparatus. And as it progresses through, the Golgi apparatus continuously sorts all these proteins out into different areas, depending on where they're going to be delivered. So this is what this part of the cell does. So you can see them being sorted out into different colours there. Now, on the walls of the Golgi apparatus, there are some security markers which latch on to that address molecule that's on the protein. So they latch on, those proteins latch on to those particular security markers. That part of the Golgi apparatus then buds off. And then it heads off towards the lysosome, which is the waste disposal unit. Now those markers also act as like security markers. So when the vesicle arrives at the lysosome, they're scanned as well. And if the lysosome is happy with it, it allows it to merge with the vesicle, and the proteins are then delivered. And the proteins can't be used yet because they're not switched on. They need to be switched on. So another process to occur to enable them to be switched on, and then they can be used in that part of the cell. And then the outer wall of that lysosome then buds off again, and those security markers are then returned to be recycled back to the Golgi apparatus. Now, I don't know about you, but I find that absolutely incredible. That's just in a cell, and that's a process that's taking place many billions of times in every cell in your body during your lifetime. And it's just one of many processes that's got to take place. When we're told, remember what I said at the beginning, that these amino acids, the building blocks of life, they're joined together to make proteins, that's what scientists assume. We can see that, in fact, amino acids, creating an amino acid, is just the starting point of an incredibly complex process. And this is just one of many such processes that must take place regularly in all living cells in order for life to continue. And if there's just one defect in this system, then the result's tragic. There's something called eye cell disease, and it's caused when there's just one defect in the process. And the protein never reaches the correct destination. It's a bit like a letter being delivered to the wrong address. And as a result, the cell cannot dispose of its waste products. And the terrible consequences that are there on the screen. And it shows just how vital it is to have all of these complex systems in perfect working order before the cell can work. Nobody has offered a plausible explanation of how these systems could have developed as required by evolution. In fact, scientists really struggle with the evolution of the cell. Here's a quote from one evolutionary biologist, a man called Ali Demersoy, who is at least honest enough to say, "I don't know." he said one of the most difficult stages to be explained in evolution is to scientifically explain how organelles, that's the different parts of the cell, developed from these primitive creatures. No creature or group has yet been found with organelles of a simpler construction in any way, or which are more primitive. In other words, the organelles carried forward have developed just as they are. They have no simple and primitive forms, which could be shortened to say, we don't know how it happened. We must remember not to confuse facts with theories and suppositions. The facts that we've just seen there are the cell is the basis of all life. It is incredibly complicated. It contains many irreducibly complex systems. And tragic results occur if just one element in one of the many complex systems doesn't work correctly. No one has ever discovered a partially developed cell. And no one has explained how the cell has evolved. It could be argued that the many irreducibly complex systems in the cell suggest a designer, a creator. It's often assumed that all scientists believe in the spontaneous chance creation of life, followed by evolution. Well, just for your information, there is a website called Descent from Darwin, and it was set up by a group of scientists who do not accept the theory of evolution as a satisfactory explanation of the complexity of life on the Earth. And scientists from around the world can apply to add their names to the list that's on this website to show their disagreement. The only criteria is that you must have a PhD in a scientific discipline, or you must be a qualified medical doctor who is a professor of medicine. And over the last few years, hundreds of scientists have added their names to the list. And despite the impression that we get about science and scientists, especially when it comes to the origin of life, there are some things that scientists just don't know. Here is a short clip of a well-known evolutionary biologist commenting on the origins of life. For those who don't know, some of the young ones may not know, it's a man called Richard Dawkins, who is very much a proponent of evolution, and he does tend to mock those who believe in God. But he was asked how life began, and here was his reaction to it.
Well, how did it start?
Nobody knows how it started. We know the kind of event that it must have been. We know the sort of event that must have happened for the origin of life.
What was that?
It was the origin of the first self-replicating molecule.
Right, and how did that happen?
I told you we don't know.
So you have no idea how it started?
No, nor has anyone.
Nor has anyone else. What do you think is the possibility that intelligent design might turn out to be the answer to some issues in genetics or in evolution?
It could come about in the following way. It could be that at some earlier time, somewhere in the universe, a civilization evolved by probably some kind of Darwinian means to a very, very high level of technology and designed a form of life that they seeded onto perhaps this planet. Now, that is a possibility and an intriguing possibility. And I suppose it's possible that you might find evidence for that if you look at the details of biochemistry, molecular biology. You might find a signature of some sort of designer. And that designer could well be a higher intelligence from elsewhere in the universe.
I played that clip not to mock Richard Dawkins, but to highlight the fact that there are limits to what science can currently tell us, such as the origin of life, and that even the most devout anti-creationists might be prepared to consider that life on Earth might have been designed. We've seen how incredibly complicated life is, even in the simplest of cells. And this is in fact just what the Bible tells us in Psalm 139. I will praise you, for I am fearfully and wonderfully made. Marvellous are your works. So, here are some points to think about. Firstly, we have seen that science has no answer to how life on Earth started. And evolution has not proved to be a satisfactory answer to the development of cellular biology. Secondly, we must remember that often People want to believe in evolution because creation is unacceptable. And third, remember that when the facts are considered, science cannot rule out the possibility of a creator. Even evolutionary scientists say it is difficult to believe that our incredibly complex, organised world happened by chance. So in conclusion, I would appeal to you, don't ever allow yourself to be deceived. Scientists make very bold statements that make it sound like they have all the answers about life and evolution. But dig a little bit further, and you'll find nothing could be further from the truth. The more scientists discover, the more questions you realise that they have. And as you've seen, they don't have a clue how life began or how it became so complex. We all have the God-given ability to understand these things. Wisdom and knowledge are not the special prerogative of people who consider themselves to be experts, whether they are scientists or theologians. And so, don't take my word for it. Have a look at the complexity of life and what scientists have to say about it. I think that the organisation and incredible complexity of life cries out that there is a creator. The heavens declare the glory of God, and the firmament showeth his handiwork. Day unto day uttereth speech, and night unto night showeth knowledge. There is no speech nor language where their voice is not heard. He causeth the grass to grow, and the herb for the service of man, that he may bring forth fruit out of the earth. When we just stand back and look at the world around us, the visible things, the invisible things, when we look at the incredible creatures that inhabit the earth, whether that is the birds that cleave the air, or the animals that hunt on the plains of Africa, or the microbes that live in the soil, when we consider the complexity at every level, even down to the smallest element of life within the cell, there is such beauty in the creation all around us. We can but marvel at our Creator, praise his works, for we are indeed fearfully and wonderfully made. Thank you very much for listening.